Mr. President and Board, and thank you for coming to today's 2030 fiscal year review. I'd just like to say that based on this current graphic, I think we should increase our bounty house spending by about 5%.
Stop! Mr. President, we are under chemical warfare attack.
What's the big deal?
Oh my god, I brought my colleague here today to tell you about it.
Thank you and hello, Mr. President. How familiar are you with chemical warfare?
Not at all.
Okay. So we're just going to go back to the very beginning, and that's World War I, where it was the first large instance of chemical warfare. Mostly what was used was tear gas, chlorine gas, and mustard gas on enemy trenches. We can show here all the chemical formulas for tear gases, chlorine, and mustard gas. And these worked as more of incapacitants rather than actually trying to kill the enemy, more just to render them useless. And now I will pass it on to my colleague to go into more depth on these incapacitants.
Thank you, Dr. Hussey, for that amazing introduction. Mr. President, I'm here today to teach you all about incapacitants. Sulfur mustard is a blistering agent, or also known as a vesicant, that causes severe, delayed burns to the eyes, skin, and the respiratory tract. Sulfur mustard damages cells within minutes of contact. However, the onset of pain and other health effects are delayed until hours after exposure. Large exposures to sulfur mustard may be fatal. Sulfur mustard has been used as a chemical warfare agent to cause delayed casualties. It has an odor of garlic, onion, horseradish, or mustard. Scientists first prepared pure sulfur mustard by the reaction of thiodicglycol with phosphorus trichloride, as you can see on reaction scheme 5.2. Thiodicglycol was prepared by the reaction of 2-chloroethanol with potassium sulfide, as you can see on 5.1. Sulfur mustard was produced for the use in warfare by what is known as the Levenstein process, the reaction of ethylene with sulfur dichloride. The fundamental reactions are the addition of sulfur dichloride to ethylene to form 2-chloroethylsulfonal chloride, as you can see in 5-3, and the addition of that compound to a second molecule of ethylene, 5-4. The relevance of DNA damage and repair to the vesicant action of sulfur mustard is supported by the observation that inhibitors of the DNA repair significantly aggravate skin injury. Sulfur mustard at neutral pH alkylates purines, pyrimidines, nucleosides, and nucleotides. The molecular basis for the high toxicity is the ability of sulfur mustard to form interstrand cross-links between guanines of the double helix, which prevents strand separation during DNA replication. Although sulfur mustard also reacts with the RNA, proteins, and phospholipids, the consensus of opinion has been that for some time that it has been the alkylation of DNA that is by far the most deadly of its actions. The enemy has attacked us with another very dangerous chemical warfare compound. This one is known as lewisite, another vesicant that contains organic arsenic instead. During World War I, a U.S. chemical warfare research laboratory investigating arsenic compounds as potential war gases developed a potent vesicant, subsequently named lewisite, after the research group director. Purified lewisite is a colorless, oily liquid at room temperature with a faint geranium-like odor. More volatile than sulfur mustard, this agent can be used as a vapor over large distances and has been mixed with sulfur mustard to achieve greater effectiveness in combat. With a freezing point between negative 18 degrees Celsius and 0 degrees Celsius, lewisite is effective over a wider temperature range than sulfur mustard. The preparation of lewisite, as you can see L1, by the original procedure is complicated and dangerous. It involves a reaction of acetylene with arsenic trichloride by using aluminum chloride as a catalyst. The reaction yields 3 principal products, as you can see up on the screen. The optimum yield of lewisite is about 20%, obtained along with the products L2, L3, tar, and an explosive material. Acetylene reacts with arsenic trichloride in hydrochloric acid solution with mercuric chloride as a catalyst to give lewisite an 80 to 85% yield. In contrast to sulfur mustard, lewisite exposure is characterized by immediate onset of pain. This agent is lethal at sufficient doses, produces a chromosomal aberrations in some mammalian cellular assays, and is a systematic poison when absorbed into the bloodstream.
Alright, so the next era of chemical warfare we're going to talk about is World War II. There was a few isolated events leading up to World War II that used kind of the similar tactics that they used in World War I with the incapacitants, and that was the Italy invasion of Ethiopia and Japan invasion of China. But once the World War II really got going, there's a shift from more of the battlefield warfare to using chemical warfare against civilians. This was most evident in the concentration camps, where the Germans used a hydrogen cyanide fumigate called Zyklon B to to kill the people in the concentration camps. This is it's called a blood agent, and my next colleague will now go into more detail of what that is.
Good evening, Dr. Higgins, recipient of the 2029 Nobel Prize.
Well, thank you. So, blood agents are poisons that affect bodily functions by preventing the normal utilization of oxygen by body tissues. Blood agents are lethal because they are fast-acting, volatile, colorless gases, and as chemical weapons, they are dispensed as aerosols and cause damage by inhalation. Due to the volatility, they are most effective in a closed area. Most common blood agents are hydrogen cyanide and cyanogen chloride. So I'm first going to talk about hydrogen cyanide. So hydrogen cyanide was first discovered in 1872 as an industrial chemical, but it wasn't until the First World War hydrogen cyanide was used as a chemical weapon by the French. While entering the body, the toxicity of hydrogen cyanide inhibits cellular oxidative processes. Humans can usually withstand 50 to 60 parts of hydrogen cyanide per million parts per hour, but exposure to 200 to 500 parts per million of air for 30 minutes is fatal. Common symptoms with small dosages are dizziness, vomiting, cherry red skin color, and just overall death. After large dosage, victims become anxious and start to hyperventilate. Death can occur within 8 minutes of exposure. So hydrogen cyanide contains a cyanide functional group. In industry, hydrogen cyanide is reacted with ketones and aldehydes to produce hydroxynitriles through nucleophilic substitution. The cyanide ion is very attracted to iron in the ferric state, forming this compound right here, and once entering the biological system, it attacks an enzyme in the mitochondria that is responsible for the utilization of oxygen in the cell. That's what makes it so dangerous, because it blocks oxygen from the body. Another one is cyanogen chloride. So cyanogen chloride is a highly volatile and toxic chemical that interferes with the body's ability to use oxygen. By 1945, the USA military was utilizing cyanogen chloride as a chemical weapon. It was seen as an effective against Japanese forces because their standard issue gas masks lacked the barriers that would provide protection against cyanogen chloride. Cyanogen chloride is pungent, heavier than air vapor, which can cause irritation of the eyes, nose, and throat. This is a distinct contrast to hydrogen cyanide, which has no irritant properties. Common symptoms include sore throat, nausea, vomiting, paralysis, and high exposure death. So, cyanogen chloride contains a nitrile function group. Cyanogen chloride attacks the body in a similar way that hydrogen cyanide does. It inhibits cells to obtain oxygen, which then causes choking. Cyanogen chloride can contaminate food, water, and air, making it very versatile in return making it very lethal. Thank you.
So the next era we're going to talk about is kind of the modern day era of chemical warfare. Most of the stuff that is being used here is mostly against civilians. We don't see much battlefield chemical warfare anymore. The two biggest occurrences that we've seen is the attempted genocide of the Kurds in northern Iraq and against rebel groups in Syria. And they mostly use a combination of mustard gas, VX, and sarin gas. And the last two there, VX and sarin gas, are what we call nerve agents, which my next colleague will introduce and go into more detail.
The first nerve agents were invented by accident in the 1930s when German researchers were trying to make cheaper and better alternatives to insecticides, but soon found that the compounds they created were deadly. Unlike traditional poisons, nerve agents don't need to be added to food and drink to be effective. They are quite volatile, colorless liquids, and the concentration of the vapors at room temperature is lethal. All nerve agents belong to a group of organic compounds that contain phosphorus, and all work in essentially the same way, by inhibiting the action of a crucial enzyme in the body that allows muscles and organs to contract. Without the enzyme's action, the muscles and organs are constantly stimulated and stop working properly. Acetylcholine is the organic chemical that functions in the brain and body as a neurotransmitter, a chemical message released by the nerve cells to send signals to other cells. The chemical drifts across a synaptic gap and triggers the next response, for example, arms moving or lungs breathing. Once the response is ready to stop, the acetylcholine pops off the receptor and is collected by the acetylcholinesterase. Once the acetylcholine has been released and the message is passed along, it needs to be mopped up by the acetylcholinesterase to prevent neurons from muscles continually contracting.
Here we see the mechanism of acetylcholinesterase with the nerve agent soman. Soman has a relatively higher affinity for the active site of cholinesterase compared to that of acetylcholine, and at the active site it undergoes rapid nucleophilic substitution between the hydroxyl group and the phosphorus right here. In the next step, fluorine leaves the molecule. Fluorine is an excellent leaving group in nucleophilic acyl substitution reactions because the departure of the fluoride in the second step is facilitated by this oxygen right here by transferring its electrons to form a double bond with phosphorus. Further dealkylation aided by water leads to the formation of this covalent adduct, which is quite stable under biological conditions, causing the enzyme to be irreversibly inactivated.
Okay, Mr. President, after we have concluded our presentation, we decided to create something that could help counteract these attacks, something we can bring to the table. And we have created this weapon, the pungent gaseous war ender, aka the PGW. And we have brought you this for you to see how colorless it is. I'm going to slowly take it out.